students, today we are going to do some MCQs from molecular basis of inheritance, and it will be whole NCERT exemplar. Okay, so let's get started. 1 minute. In a DNA strand, the nucleotides are linked together by phosphodiester bonds. Okay. A nucleoside differs from a nucleotide as it lacks the phosphate group. Okay. Both deoxyribose and ribose belong to a class of sugars. Called as pentoses. Okay. The fact that a purine base always paired through a hydrogen bond with a pyrimidine base leads to in DNA double helix uniform width throughout the DNA. Okay. The net electric charge on DNA and histone is negative and positive. DNA is negatively charged and our histone is positively charged. Okay. So while electrophoresis we use this property of DNA. Okay. The promoter site and the terminator sites for transcriptions are located at 5 upstream end and 3 downstream end respectively of the transcription unit. Okay. Which one of the following statement is most appropriate for sickle cell anemia? So, all of the above, as how we can see that it cannot be treated from iron supplements. It is a molecular disease. It confers resistance to acquiring malaria. Okay. And in African countries, it is resistant to malaria, so useful. Okay. One of the following is true with respect to AUG. So, what is importance of AUG? As it is a start codon, so it codes for methionine only. It is also an initiation codon. It codes for methionine in both prokaryotes and eukaryotes. So, answer is all of the above. Okay. The first genetic material could be, as we know that RNA is the first genetic material and it is the most primitive one. So the answer goes to RNA is the first genetic material. Okay. With regard to mature mRNA in eukaryotes, it is speaking about mRNA in eukaryotes. So you can do this question by exons appears but introns do not appear in the mature RNA as it goes under splicing. Okay. The human chromosome with the highest and least number of genes, in them are respectively chromosome 1 and Y. Chromosome 1 is having Who amongst the following scientists had no contribution in the development of the double helical model for the structure of the DNA? So the answer goes to Michelson and Salt. They have done the experiment on the semi-conservative natures of the DNA, as this is said by two scientists who discovered DNA, and it is not the Michelson and Salt. They have only done the experiments on the E. coli bacteria and not discovered the semi-conservative nature. Okay. DNA is a polymer of nucleotides which are linked to each other by 3 and 5 phosphodiester bond. 3 to 5 phosphodiester bond to prevent polymerization of nucleotides. Which of the following modification would you choose? So our answer goes to remove or replace 3 OH group in deoxyribose. Okay. Discontinuous synthesis of DNA occurs in one strand because DNA DNA polymerase catalyzes the polymerization only in one direction, that is 5' to 3' or 5' to 3'. Okay. Which of the following step in transcription is catalyzed by RNA polymerase? Transcription is catalyzed by RNA polymerase is elongation. Okay, elongation is catalyzed by RNA polymerase. Only elongation. There are 3 steps, but only elongation is catalyzed by RNA polymerase. Okay. Control of gene expression takes at the level of takes place at the level of transcription. Okay. Regulatory proteins are the accessory proteins that interact with RNA polymerase and affect its transcription. Which of the following statement is correct about regulatory proteins? So they can act both as repressor and what? They can act both as activators and repressors. Okay. Which was the last human chromosome to be completely sequenced? So the answer goes to chromosome number 1. In May they have sequenced it, and it is the last chromosome to be sequenced. Okay. Which one of the following are the functions of RNA? Functions of RNA. So the function of RNA is the carrier of gene information from DNA to ribosome synthesis and peptides. It carries amino acids to ribosomes and it is constituents composed of ribosomes. So the answer is all of the above. Okay. While analyzing the DNA of an organism, a total number of 5,386 nucleotides were found, out of which the proportion of different bases were adenine 29%, guanine 17%, cytosine 32%, and thymine 17%. Considering the Chargaff's rule, what Chargaff's rule we have to consider? Then the answer includes is that. So the answer is it is a single stranded DNA. How we can find it? As you know that A pairs with T and G pairs with C. So here you can see that adenine is 29%. So the guanine, so the thymine should also be 29%. What you are seeing here that it is what? It is 17 and guanine is here. 1 minute. Guanine is here 17 and cytosine is 32. So we can say that it is a single stranded DNA and they are not complementing each other's activities. Okay. 1 minute. If Michelson and Salt's experiment is continued for 4 generations in bacteria, the ratio of 15N upon 15N is to 15N upon 14N is to 14N upon 14N containing DNA in the 4th generation would be 0:1:7. How I can prove it for all of you? 1 minute. If the sequence of nitrogen bases of the coding strand of DNA in a transcription unit is 5 ATGA8TG3', the sequence of the bases in RNA transcription would be. So the answer is A, that is 5 AUG. How? Let's see. So here you can see that The sequence of nitrogenous base in transcription unit is this one. Okay, so that 5 ATG AATG. Okay, so it is asking us the what would be the sequence in RNA transcript. So transcription unit is having this. So when we convert it, as you know that for A it comes U. Okay, so as you know that it is our template and it is our coding strand. So the T for template and T for 3, and so you can learn it by 1 minute. This, okay, so this is template strand and this is coding strand, which is having 5 prime to 3 prime. Okay, so it is a given is for coding strand, so we gonna do, as you know that template strand is being transcribed, or so we will write 1 minute. The coding strand is having 5 and 3. So what you have to do, wherever T is written, you have to convert it by U. Okay, and your answer will be found like this. Here ATG is written, so your answer would be AUG. So our answer would be A. Okay. In the RNA polymerase halo enzyme, 1 minute. The RNA polymerase halo enzyme transcribes the RNA polymerase halo enzyme transcribes the structural gene and the terminator regions. Okay, so the answer is C. If the base sequence of a codon in mRNA is 5 AUG 3 prime, the sequence of tRNA pairing with it must be 5 CAU. How? I will show you. That the base sequence of codon is 5:3 AUG. Okay. So what we gonna do is 1 minute. So 5:3 is AUG. So here will be 5 CAU. Okay. So our answer is this one, and it matches with this UAC 5:3. Okay. So this is the answer, as it is asking for tRNA pairing. So the codon anticodon is UAC 3 prime to 5 prime. Okay. The amino acid attaches to the tRNA at its what? Its third end. Okay. To initiate translation, the mRNA first binds with the smaller ribosomal subunit. So when this is, and whenever translation occurring, then mRNA passes through this smaller ribosomal subunit. Okay, so this is the answer. To initiate translation, that mRNA firstly binds to the smaller ribosomal subunit. Okay. In E. coli, the lac operand gets switched on when lactose is present and it binds to the repressor. So the switch is on when the lactose is present and bind to the repressor, as lactose will form the glucose and it will lead to the functioning of the metabolism. So when it is switched on and when it is switched off, whenever the repressor is absent, so it will be switched on, and whenever repressor is present, then it will be switched off. In the presence of lactose, lactose binds with the repressor and it prevents binding of the inhibitor to the RNA polymerase. So transcription or translation occurs and the metabolism goes on. Okay, so we can learn it by IPOZYA, Z for beta galactosidase, Y for permease, and A for transacetylase. Okay, all the best for NEET 2021.